Okay.
Okay, now we can see it.
Okay, perfect.
Yeah, you're good.
Okay, so I did algae. So algae are simple photosynthesis eukaryotes, and each type contains chlorophyll and other accessory pigments that can absorb different wavelengths of light for photosynthesis. So there's different types. So the first one that I chose was diatoms, which means cut in half. They are the most abundant type of algae because they are found in saltwater, freshwater, and even terrestrial environments. They are also distinguished by their peristyle, which are their like external structures, which can also mean that they're called jewel of the sea. And they usually have a golden brown color because of the pigment called fucoaxanthin that is abundant enough to make other pigments. They're also single-celled. So the other algae is called brown algae. They are a large group of multicellular algae that are usually grown in cold salt water. Sargassum, however, is the only exception because they grow in warm saltwater. So a common type of this would be like kelps because they could grow up to 75 meters long and they are known as the underwater forest. They also contain the same pigment as diatoms because it also gives it a brown color. So these ones are also commercially valuable because they are the source of alginate, which is needed for like ice cream, toothpaste, etc. And then we have green algae. They are the most single-celled, while some are multicellular. They can grow in freshwater, saltwater, like on rocks, trees, and like more terrestrial surfaces. So they get the green color from chlorophyll, but they can be other colors as well. An example of this will be volvox, and they can form colonies. So then we have red algae. They can be both multicellular or single-celled, and they are found in warm saltwater areas like tropical coastal areas. They have a reddish color due to the pigments called phycoerythrin and then phycoxim. Sorry, I can't pronounce it, but they allow it to grow in like water like very deep, like 70 meters. And then these are also commercially useful. For example, like a group of red algae called Coraline's secret, like calcium carbonate that is used for bone grading with both medical and dental. They're also the source of gelling agents, which is agar and carrageenan. And then we also have another type, but it's not really considered a type, which are called omeocytes, which are water molds. They are both like closely related to brown algae and diatoms, but they cannot like photosynthesize and they lack a chloroplast. So how algae reproduce is they can reproduce both sexually and asexually. When they can produce sexually, they can alternate between haploid and diploid generations. So algae are medical significance because they contain anti-inflammatory compounds like omega-3 polyunsaturated fatty acids and chlorophyll A that can be used to treat many diseases like cardiovascular diseases and anti-inflammation metabolic diseases. Sorry.
Okay, and then I had protozoas. So like what are protozoa? They're more easily defined by like what they're not because of their diversity. And so a good way to define them would have to be heterotrophic unicellular eukaryotes that are not fungi. They're distinguished and they're called animal-like and most lack a cell wall. And many have specialized cilia, flagella, or pseudopods, and that's basically like how they get around, so that like determines their movement and stuff. And several types lack mitochondria, and they are an important part of the food chain, so they devour bacteria and algae, and are also food for like larger species. And the majority of protozoa are free-living aquatic organisms that are essential to decomposition of materials such as zooplankton. That's the background of the slide. Um, and then types of protozoa. The first one that I have written down is the trophozoites, and that's the growing form, and they grow and exist within like the intestinal tract. And then they develop into like cysts as they exit through feces. And the cysts the cysts refer to the infection survival form. And typically they're hardier and can withstand and survive like a harsher environment. And then the oocysts are going to be an environmentally resistant form that are produced by a group of intestinal protozoa called coccidia, and these may require time outside of the host to mature, and that is the primary difference between them and the cysts. And then for my second one, I have amoeba or amoebae. And they move by retraction and extension of their cytoplasm by means of pseudopods. And again, that's like the fake feet, and that's exactly what it stands for. And that's how they get around. So the cell shape changes as it moves because of the lack of the cell wall, and then they reproduce via binary fission, so that's asexually. And then the intestinal amoeba can form cysts. And then I have a couple of examples there. It's the entamoeba histolytica. I hope that's I said that right. And that's the pathogen that causes disease and that results in dysentery. And then Naegleria fowleri that invades and destroys brain tissue via access to sinuses, and they live in free-living warm bodies of water. So if you've ever heard like of a brain-eating amoeba, like when you go swimming, like that's probably the one that you've heard of. And then for my third one, I wrote ampicomplexans, and then the medically important types are obligate intracellular parasites and require host cells to replicate. And basically, apical complex aids in the penetration of the cytoplasmic membrane of host cells. So that's just something that helps them to essentially get into it. And then they have complicated reproduction cycles because they utilize both sexual and asexual, but they also require multiple fission. So an example, Plasmodium species cause malaria. So in 2018, about 228 million people suffered from malaria, and there were about 405 recorded deaths. And the sexual reproduction portion occurs in the definitive host, and then malaria is in malaria, the definitive host is going to be that mosquito, and then the intermediate host is going to be that human. And then I have a couple of pictures on the next slide. And then here it just shows like the transmission to like human, and then the sporozoites like entering the liver and infecting the hepatocytes, and then the liver cells go ahead and rupture, and then the merozoites are released. And then from there, the intra-arthrotic cycle starts, and then the sexual cycle, and then that like starts all over again to the transmission of another mosquito. So it really just affects your blood cells, and it's a vicious cycle because it goes from mosquito to mosquito and then mosquito to human, and it's just really a really long cycle. And then I have a couple more types of protozoa. For number 4, I wrote the hemoflagellates, and then they are basically flagellated protozoa that multiply by binary fission. And they live in the bloodstream for part of their life and require a blood-feeding insect for that portion of the cycle. So that can be like mosquitoes, that can be bed bugs, that can be kissing bugs. And the kissing bugs is the insect that I used for my example. So the T. cruzi is transmitted by kissing bugs and causes American tri trypanosomiasis. And that's basically what we like to call Chagas disease. And then number 5, I wrote lumen-dwelling flagellates, and they live in the lumen of the intestinal or genital tract, and they use flagella for movement. And Giardia lamblia is a flagellate that lives in the intestinal tract and causes diarrhea. And then here there's just like a list of like pathogens and their characteristics, and then the diseases that they give. You can go ahead and like pause the video or pause the presentation if you want to look at it. There's a couple of examples that I put in there that were also part of the slide, so I went over that a little bit, but there are other protozoan groups such as slime molds, social amoebae, which are the same thing as cellular slime molds, and then plasmodial slime molds, and then euglenids and dinoflagellates. And the thing about dinoflagellates are they have been known to produce neurotoxins in like tides and stuff, and that's also known as red tide. And this happens when blooms in like tide produce brevetoxin, and it affects the shellfish and fish. And this can lead to diseases such as NSP and then PSP when consumed. And basically, it just makes you really sick when you eat shellfish. And um, that's how the dinoflagellates are medically important. So just a little bit of more medical importance for protozoa. So as seen in the slides prior to this one, there are many diseases caused by pathogentic protozoa, whether they be through amoebas, through ampicomplexans, or any other types. However, there are many protozoan species that are both ecological ecologically beneficial as well as beneficial to our health as humans. And in the medical field, it's important to be aware and up to date on the symptoms of these diseases, as well as up to date on how to treat them, because structure determines function. And we have to understand what we learn about these organisms in order to know how to handle them and manipulate them.
Yeah, so slime molds are very similar to fungi, as in they have similar spore creation, but they're not classified as such because of their amoeba-like gliding movability. So basically, they move differently. There's also two types of them, one of them being cellular slime molds, which are just single cells of like bacteria that ingest other bacteria and live on decaying vegetation like dead logs, trees, bushes, leaves, whatever, that can turn into what's called slugs, aka just clusters of fruiting bodies that contain spores. Just like the picture on the right with all the orange, that is all the fruiting bodies, which would be called a slug. The second kind is known as plasmodial slime molds. Now, these are large, like very large, multi-nucleated slime molds that just come in bright colors, like the picture on the right with the yellow on the log. Their life cycle typically involves having fruiting bodies that contain spores that once they contact some organic matter, also like logs or trees or leaves, they just start to reproduce and they start to create what's called plasmodium. And this plasmodium is like a slime that just oozes all over the log, like what's seen on the picture. And it just eats everything in its way. It just eats microorganisms, debris, anything. And once it starves, it starts to create the fruiting bodies again, and then the cycle repeats itself. Another type of mold is water molds, aka oomycetes. And these are fungi-like organisms that live in just really moist areas. They can even live under the water. And they tend to be parasitic. They have caused diseases such as downy mildew, pythium, and Phytophthora root rot, which have in the past, in history, have caused famine in many ancient places, or not even ancient, just in the past. What they like to do is they absorb food from their surrounding environment. And if there is no food, but they find an organism, they go into the organism and just start eating it from the inside out. The picture on the right shows a fish that has been infected with such water mold, and its eye has pus coming out of it, and it's just infected and not good to eat, will cause food poisoning and other such complications.
Okay, once again. Okay, so viruses in human tumors. Virus has the power to cause cancer. Some examples are hepatitis B, that is that liver cancer, Epstein-Barr virus, that is the Burkitt's lymphoma, and HPV. Viruses can cause a cancer in many ways. Some of them are when they associate with host proteins, they also proliferate the human immune system when it's weak, and hijack those human cells. Also, bacteria impacts viruses and tumors. A study that was conducted in 2018, it was found that infections are estimated to contribute to 20% of all human tumors. They also seen that they have also been seen inside of tumor cells, which increases one's risk of getting cancer. So they not only like cause that cancer, but they can like live inside of of like those tumor cells, which make it even more risk. And something that is so important to take into account is know what we eat and what we put in our bodies, because this can really like help us to like prevent that cancer.
So cultivating bacteriophages is basically, bacteriophages are viruses that infect bacteria and replicate within bacteria. Cultivation is basically involving the growing of bacteriophages in bacterial cultures. We start off by isolation, which means obtaining a sample suspected to contain bacteriophages, such as sewage or water from environments where bacteria thrive. Next is the enrichment, which is mixing the sample with the host bacterium susceptible to bacteriophages to allow the viruses to infect and replicate. Next, we do plating, which is applying the enriched sample onto a bacterial lawn on an agar plate to form plaques, which are areas where bacteriophages have lysed bacterial cells. Next, we do the picking plaques, which is selecting the individual plaques from the agar plate and transferring them into a medium for further propagation. Next is amplification, which, as we know, is the growing of bacteriophages in liquid culture to obtain a higher concentration for subsequent experiments. Quantitating bacteriophages involves determining the concentration or number of phages presented in the sample. Methods for this include the plaque assay, which means counting the number of plaques formed on the actual plate to estimate the concentration. Another form would be direct counting, which is done by using an electron microscope to visualize and count individual phage particles. Next would be optical density, which is measuring the turbidity of the phage suspension using a spectrophotometry, which correlates to the concentration. Benefits include the understanding of bacteriophage biology, which helps with researchers studying the morphology, lifestyle, and interactions of the bacterial hosts. Phage therapy is helps with the potential applications in treating bacterial infections, and environmental monitoring helps with providing insight into microbial populations and ecosystem dynamics. Cultivating animal viruses, we start off with, again, the animal cells growing in culture discs or flasks. Viruses are introduced to the cells, allowing them to infect and replicate within the cells. Incubation is next. This is where the infected cells are kept in a controlled environment, often in an incubator set to the optimal temperature and humidity in order to help with the virus growth. Next is the observation, which is where we monitor the cells for signs of viral infection, such as changes in the cell morphology or the appearance of viral particles under a microscope. And then harvesting includes the infected cells getting harvested once the observation is done and they're isolated from the cells. Quantitating animal viruses, again, would be the plaque assay, which are the infected cells that are mixed in the semi-solid medium and plated. These are the viral particles that cause the visible plaques or clear zones on the cell monolayer. The number of plaques is counted to estimate the viral number. Some benefits from this would again include understanding the viral biology, which helps scientists study their behavior, the replication cycle, and interactions with host cells. This helps with vaccine development, antiviral drug testing, and then disease diagnosis, which helps diagnosing viral infections and assessing their severity.
All right, and when tying all this info back to the Christian worldview, we wanted to highlight Philippians 2:3-4, which states, do nothing out of selfish ambition or vain conceit. Rather, in humility, value others above yourself, not looking to your own interests, but each of you to the interests of others. So we definitely can apply that verse to the Christian worldview to science careers, whether that be in the hospital, the clinics, the labs with our peers. And we want to tie that to three main points of human value and dignity. One being every human, every student, every person is created in God's image perfectly. And two, that ties into therefore every person has intrinsic value, which therefore means each person's value should be upheld in research and practices. And then if we also want to tie the Christian worldview into the application of microbiology in this sense, one of the first points is understanding the complexity of God's creation, which we all know is very vast. And the verse Psalm 104, 24-25, that verse praises God's creation, both big and small, as we've been come to see. Understanding his creation under the lens of microbiology, whether that be algae, protozoa, slime molds, viruses, etc., allows us to celebrate him and see his true craftsmanship as our God and our creator. And the second point I wanted to highlight was us being stewards of his creation. Genesis 2:15, while it doesn't state it there, it states, the Lord God placed the man in the Garden of Eden to tend and watch over it. And how does this relate to us and the modern world? It's showing that God puts us, in the Garden of Eden, which is the world, to take care of it and nurture it, and therefore be stewards of earth to it and respect it. So, but in order to take care of his creation, we all must understand all that is in it and recognize its connectedness.
All right, and then to kind of finish off what Christopher was saying, I wanted to talk about the ethical practices that we can apply the Christian worldview to in microbiology and other science careers. As stated on the previous slide, there are many Bible verses that go into depth about the importance and the dignity of human beings and God's creation. So the biggest thing to take away from that is to not be selfish with your practices and to be mindful of the things that you're doing, whether it be with another human being or the creation that God has made for all of us. And you can do that in several ways by infusing biblical methodologies into practices. And so we have three different ways that you can do that. The first being integrity and honesty, the second being respect for life, and the third being community and collaboration. So Proverbs 11 verse 3 talks about integrity and honesty by saying, honesty guides good people, dishonesty destroys treacherous people. So whenever you're in the practice and doing things like that in microbiology or other scientific careers, you just want to ensure the honesty in everything that you do. Second is respect for life. The psalm that was placed here while the quote isn't directly on the screen, it glorifies God because he made everyone intricate and unique. And so we must treat each other with the respect that God has made us perfect and unique, and that we all have intrinsic value because of that. And then the third, community and collaboration, from Ephesians 4:16, talks about that in order to succeed, we must all grow together as one body in Christ, which is what God intended it for us to be when he created us, to all come together and work together in community. And so the first thing in order to do that is to love each other, to continue to grow both inside the practices of microbiology and outside. So lastly, we want to commit to transparency, integrity, and honesty in the practice, just to continue to research and practice and learn more about God's creation in a microbiological sense, as well as the creation of God too.
And these are our references. Right.